while the room gets settled for this next section on extracellular vesicles, I'm Raymond Schiffelers. I'm the chair of this session. I work at the University Medical Center in Utrecht, and I have a group there that works on nanoparticles. Lipid nanoparticles, basically, and both biological as well as synthetic lipid nanoparticles. And in this presentation, I will compare them head to head. If everybody's settled, we can start with the first slide. There we are. So today we're going to explore a head-to-head comparison of synthetic lipid nanoparticles and biological extracellular vesicles for the delivery of RNA. And the interest in extracellular vesicles became quite major over the last 20 years, I would say, where they might represent a class of natural lipid-based nanoparticles that could exchange materials between cells. And it seemed like a natural nanoparticle that could deliver basically any molecule that you can find inside a cell, because proteins, RNA, DNA fragments have been recovered inside extracellular vesicles and could be exchanged between cells. And this could perhaps be exploited therapeutically, and people are claiming that because they're more natural than, for example, synthetic LNPs, they could have benefits in, for example, adverse effects. On the one hand, it's very intriguing that there are so many different biological molecules in there. At the same time, it means that there are thousands of different classes of bioactive molecules inside a single extracellular vesicle. So whenever you want to interpret the therapeutic effect or any effects on cellular phenotype that is caused by the exposure of these EVs to these cells, it's very difficult to think back what was responsible for this. That's a major challenge in this field. And a second major challenge is actually getting enough material, because if you have 20 flasks, you have about 10 to the 12 particles, and we'll see later how much easier it is once you switch to synthetic lipid nanoparticles. But limited amount that you can get, and then you need to isolate, and then all these difficulties apply. The donor cell dictates the composition, so if you switch donor cell, you have a very different extracellular vesicle composition. The moment you change something in the culture conditions, your extracellular vesicle composition will change, and different isolation protocols isolate different subclasses of extracellular vesicles more preferentially, so it's difficult to standardize your product. Anyway, to be able to link a cellular phenotype to, in this case, RNA delivery, we developed a reporter system that is based on CRISPR-Cas, and it works like this. So inside the reporter cell, there's this construct that expresses mCherry, so it's a red fluorescent cell, that additionally turns green if a single guide RNA arrives. So the cell has all the machinery for CRISPR-Cas, but the moment the single guide RNA arrives, there are cleavages induced, and those are repaired. And if there's a 1 nucleotide or 2 nucleotide frame shift, there's a green fluorescent protein production. So it's a clear-cut mechanism. If the cell is red alone, then nothing happened. If there's a green fluorescent protein expressed, then the single guide RNA must have been delivered successfully. You can do that, and that's panel C. You can do that with lipid nanoparticles, add the single guide RNA, and then essentially all of your cells turn green. So the system is really responsive to single guide RNA delivery. And the idea is we have a donor cell that expresses the single guide RNA. It's added to the cells that are the reporters, and then we count how many cells turn green. That's what we did in co-culture, and you see that it's already quite challenging to observe, because it shows that about 0.2% of the cells turn green if we have reporter cells and donor cells in co-culture. So they're in the same well, exchanging material together, and apparently part of that material is single guide RNA. And then 0.2% of these cells turn green, and the more donor cells you have, the more cells turn green, which sort of makes sense. And you can visualize that. If you do that without cell-to-cell contact, so you have a transwell system where you have the donor cells above that rain the extracellular vesicles on the surface of these reporter cells, we go down another order of magnitude. So now we're down to 0.03% of cells that actually turn green. But you can see that there's functional transfer of a single guide RNA that is raining down on these on these cells. And it's responsive to inhibitors of extracellular vesicles, so GW4869 is one of those inhibitors, and if you add that, then indeed the number of green cells goes down. Then the proof of the pudding, essentially, isolate the extracellular vesicles via column chromatography, characterize them first of all, so there are about 100 nanometer particles. They contain all the markers that are there for extracellular vesicles. They look like extracellular vesicles under the microscope. They contain RNA. They're taken up by cells, and if you add them, then indeed you also see green cells appearing. I must say that we put an entire flask of cells and take the EVs from that and put that inside a single well. So you could say that the odds are a bit off in a sense that it's a lot of cells that actually produce a limited amount of EVs, but then at least you can observe that green cells are being formed. So in principle, this system works, and we can demonstrate that extracellular vesicles can transfer RNA from one cell to the next. And we can identify the different pathways that are involved, such as, for example, the integrin beta-1. That is clearly, if you knock that out, then the efficiency of the process drops dramatically. But now on to the head-to-head comparison. So we took synthetic lipid nanoparticles based on MC3, loaded with, in this case, the single guide RNA. And just to annoy people that are in the EV field, so whenever I gave the presentation in an EV society meeting, then I say, well, and then you press the button, and 30 seconds later, you have 10 to the 14th particles, whereas they have to culture 2,000 flasks in order to get the same material, so just to annoy them. They're perfectly characterized, of course, a very limited size distribution with. And then characterizing them and comparing them to the biological EVs, in this case harvested from 2 cell sources. The EVs slightly larger than 100 nanometers, the LNPs slightly smaller, zeta potential, and then the characterization of the different surface proteins that are present on the EVs. Then we quantified how much RNA was actually inside, and we made a line, and we calculated the standard line for the RNA. We calculated the number of particles, and some people say, shouldn't it be the other way around? But it's particles per copy of single guide RNA. So it's about 1 million particles for one single guide RNA to be encapsulated. So 999,999 are empty, or no, not empty. They contain all kinds of things, but not the single guide RNA that we're interested in. So it's not very high. And this is in a system that is overexpressing the single guide RNA. So it's really boosting the system to the max, and then we have these low amounts. And of course, with the lipid nanoparticles, you get perfect encapsulation, and you can simply titrate that with the amount of particles that you do. Do these particles work? So we tested how much material is actually leading to this functional change in the cell, so how many cells turned green. And with 40 microliters of our EVs, we see this 0.03% response, and the same for the other type of EVs. So compare that to LNPs. That must be difficult, because we either need to titrate, so add very little, or spike in a lot of LNPs that do not contain the RNA of interest. We tested both approaches, and then you can see that we see the green bars show where there's a significant change in the number of green cells. So that's reached at concentrations at about 1,000 femtomolar, whereas the EV concentration range where this phenomenon occurs is about 1,000-fold lower. So we have already these changes. Let's go back. So here, these 40 microliters translates to the gray area in this graph. So at 1,000-fold lower concentrations of single guide RNA, EVs are already able to turn cells green, whereas we can only do that with 1,000-fold higher concentrations of synthetic lipid nanoparticles. And well, that's very intriguing. At the same time, it's not more than that, in the sense that, so lipid nanoparticles, very reproducible to make, scalable, fast, cheap, only 4 excipients. But EVs are 1,000-fold better, but at the same time, how to scale them, how to characterize them, how to produce them at scale, well, all these kind of things apply. So I think it's an intriguing observation that EVs can outperform the best synthetic lipid nanoparticles that we currently have for small RNAs. And at the same time, now comes the difficult task of trying to understand how they do that. And these are the people involved. And with that, I thank you. And we immediately go to the next speaker. I would say thank you.